I'm Peter Vekesland. I'm a professor at Virginia Tech, and it's my pleasure to be co-chairing this afternoon's session with Sucrier Sekekal, who is affiliated with McGill University here. But I will be introducing our first speaker, and then she will be taking over after that, as we've kind of been doing today. But our first speaker is my longtime friend and former member of my Ph.D. committee back in the day, Pedro Alvarez, who is the George R. Brown Professor of Civil Environmental Engineering at Rice University. At Rice, he serves as the director of the U.S. National Science Foundation Engineering Research Center on Nanotechnology-Enabled Water Treatment, or NEWT, and he now is the director of the Rice Water Institute. Pedro's research interests include the environmental implications and applications of nanotechnology, bioremediation, the fate and control of toxic chemicals, water footprinting of biofuels, and antibiotic resistance control. Now, Pedro actually is returning to Montreal because he got his B.Eng degree in civil engineering here at McGill, and then got his MS and Ph.D. degrees at the University of Michigan in environmental engineering. Over the course of his distinguished career, Pedro has received many different awards. Amongst those, he is the 2012 Clarke Prize, Clarke Water Prize laureate, and he's the recipient, most recently, of the 2023 Outstanding Achievement Award from the China Chemical Society. A number of past honors include that he was the president of the Association of Environmental Engineering and Science Professors, or AAEESP. He's received the AAEESP Frontiers and Research Award, the Water Environment Federation McKee Medal for Groundwater Protection, and has received numerous Best Paper Awards with his students. Pedro currently serves as an executive editor for the journal Environmental Science and Technology, and he was elected to the U.S. National Academy of Engineering for his practical and pedagogical contributions to bioremediation and environmental nanotechnology. And with that, I'd like to have Pedro come to the stage.
Well, it's a great pleasure to come back to Montreal. I want to start thanking Dominique for this invitation and Ed for initiating this very important conference. As you know, wastewater treatment plants do a great job of protecting public health and mitigating the spread of infectious disease. But as we heard earlier today, some wastewater treatment plants serve as breeding grounds and point sources to the discharge of multidrug-resistant bacteria that we often call superbugs. And earlier, we had a lot of great talks that advanced the molecular and evolutionary basis of antibiotic resistance that have described very well the problem. And to complement that, I would like to offer some solutions to this problem. Solutions that are consistent with existing infrastructure that don't require major infrastructure modifications so that they can be more easily scaled up and implemented. So we began this research with one question. Are superbugs reaching sewage treatment plants? And if so, how do they behave? What's their fate? What units in the treatment train remove them or grow them? And to answer that question, which was several years ago, before people were interested in funding this type of work in the US, we went to China, and in collaboration with Nankai University, we surveyed 2 wastewater treatment plants near Tianjin. Tianjin is sort of like the port of Beijing, 2 hours drive east, and they discharge to the Haihe River. And we sampled every unit in the treatment train. In this case, we were interested in one gene, the NDM1 gene, which, as you know, codes for the New Delhi betallolactamase. And we sample again the influent and effluent of the primary settling tank, the aeration tank, the secondary tank, every unit in the treatment train. And because we were also interested in not just the total, but also the relative abundance of this gene in the community, we also quantified a generic biomarker for the total bacteria, right? And well, to cut through the chase, we found that for every one of those genes that were coming into the plant, there were 5 coming out. So in effect, this plant was a breeding ground. And most of the discharged genes, first of all, they were growing, as you might expect, in the aeration tank, in the bioreactor that is like a luxury hotel for bacteria where they proliferate and mate and grow. And most of them were being discharged in the biosolids that happened to be applied to agricultural fields, raising potential concerns about food security. But a significant amount of these genes were coming out in the effluent that is discharged to the river, even though this effluent is disinfected. In fact, they were chlorinating. I don't know why, because as you know, chlorination generates trihalomethanes and other things for wastewater. But anyways, the chlorination process will remove about 90% of the total bacteria, but only about 43% of these NDM1 carriers that happen to be resistant to oxidative stress by chlorine, nothing to do with antibiotic resistance. But this is a concern because that means we were discharging free DNA, free antibiotic resistance genes, as well as live bacteria that when they die, they release these antibiotic resistance genes. And this external DNA gets stabilized by clay in the sediments of the Higher River. You can see those black bars means that they stay and persist for several weeks. In separate work, I forgot to include a slide, we showed that this free DNA can transform indigenous bacteria that essentially ends up augmenting the environmental resistome. So this underscores the need to mitigate the proliferation and discharge of these antibiotic resistance genes. And so we ask the logical question, what can wastewater treatment plants operators do to mitigate the proliferation of superbug genes? And keep in mind that a lot of these genes are present in mobile genetic elements, including resistant plasmids. And the bacteria don't like to carry excess luggage. They don't like to carry plasmids or genes that they don't need, which imply a high metabolic burden, unless they are needed. So that if you don't have antibiotics to defend against, if we were able to mitigate the presence of antibiotics at sublethal concentrations, you will remove that selective pressure to maintain these genes, and hopefully they would dump them. And we showed that a long time ago in 2004 in this paper, where this actually was the first paper that called out the need to consider antibiotic resistance as an environmental pollution problem 20 years ago. And what we were looking here is asking the question, what happens to soil bacteria when they're subjected to drainage from animal farms that carry tetracycline with it? And upon long exposure, about 20% of the, this is before omics, we were doing culturable plates, about 25% of the total heterotrophs actually became resistant to tetracycline. But when we removed tetracycline from the influence of this flow-through soil column, this appearance went back to background levels, right? So supporting this trend, our survey of the wastewater treatment plants showed that the higher the presence in the x-axis of residual antibiotics, again at sublethal levels, for two types of antibiotics, tetracycline and sulfonamide, the relative abundance of resistant genes increased. There was a significant correlation. And you may say correctly, well, correlation doesn't imply causation. Yes, there may be other factors that select for antibiotic resistance. Maybe you have heavy metals that select for efflux pumps that happen to be also antibiotic resistance genes. But my point is that when it comes to antibiotics, I think Amy did a very good job earlier today of showing that even during the pandemic, higher exposure to antibiotic does select for resistance, and Friedrich Nietzsche, therefore, was right when he said that whatever doesn't kill you makes you stronger. So what this implies is that it is very important to minimize the use, misuse, and abuse and discharge of antibiotics to wastewater treatment plants to curtail the proliferation of these superbugs in these facilities. Of course, this requires a lot of help from policy and education. Maybe that's the only thing that is going to really move the needle fast. But technologically, we can do things about it. If we can identify hotspots, point sources, maybe an antibiotic resistant factory, we can segregate that waste and use an advanced oxidation process in situ to destroy them. Another thing to remember is, again, hindering the ability of resistant bacteria to harvest energy. I'm talking about harvesting metabolic energy, exacerbates the metabolic burden associated with resistant plasmid maintenance. So especially when, again, that plasmid or that gene is not needed. In this example, we're looking at a facultative E. coli that carries a resistance gene. This, because it's facultative, can live under aerobic or anaerobic conditions. If we subject this E. coli under anaerobic conditions, which thermodynamically are less feasible to harvest and make ATP, then they begin to dump the plasmid, right? So what this implies is that maybe you want to consider having longer retention times in your anaerobic digesters. Maybe your agricultural drainages should go through anaerobic lagoons before you discharge them to, again, promote the curing of resistant plasmids. So overall, operators may manipulate many master variables of the activated sludge. They may adjust their food to microorganisms ratio, perhaps increase the wastage rate so they promote faster growth, and with faster growth, plasmids get cured, and of course, prolong anaerobic digestion time. Now, what I suspect most of you came to see was to hear a little bit about phages, right? Which are viruses that exclusively infect bacteria. They cannot infect humans or insects or protozoa or animals. So from that point of view, they're safe to humans. And they are roughly about 10 to the 31 on Earth, or roughly 10 phages for every bacterium on this planet. That means that phages are by far the most numerous, most diverse, and I would argue most underutilized biological resource in our planet, okay? This guy here is Félix Derel. He lived in Montreal, and he was one of the pioneers of phage therapy out of the Eliava Institute in Georgia, in Tbilisi, Georgia. Back then, you know, they were using phages to fight pathogens, I view phages as self-replicating green biocides that give you the opportunity for highly precise microbial control without the collateral damage that chemical disinfectants cause. You know, chemical disinfectants will ruin your reverse osmosis membranes or produce carcinogenic byproducts, okay? So phage therapy, as you know, I was mentioning, was very popular in Eastern Europe back then, and now Western countries are revisiting this forgotten cure due to the emergence of all of these difficult to treat infectious diseases, and there are many products commercially available already based on phages that have been approved by the FDA, either to, I don't know, to kill streptococcus or enterococcus, or to fight plant disease as well. But to be honest with you, using phages in wastewater treatment plant is a lot more difficult. It faces many important challenges that I want to be honest with you about. One challenge is that phages are very specific. Most phages that we know are highly selective, and one phage will infect one bacterial species. And that works well when you know what you're after, like Klebsiella pneumonia. You know, you have one enemy. But in wastewater treatment plant, where you're trying to control this inactivity that might be caused by hundreds of different species, some of which we don't even know who they are, then that becomes a real challenge. That challenge favors broad spectrum chemicals, right? The second thing is that we don't always know who the target is. You need to identify them, which is getting easier with omics. But even if you identify them, to grow the phages, you need to have the target isolated so that you can grow them on that host. And many of these bacteria are unculturable. Some of them are not susceptible to phage attacks. And on top of that, some of them might be pathogens that poses a serious occupational hazard. And finally, there are other factors. If your bacteria are in biofilm, well, phages have a hard time penetrating them, just like antibiotics do due to diffusion limitations. Some bacteria become resistant to phages. I'll talk a little bit about that later. But also, you need relatively high concentrations of your target so that you can have high concentrations of phages and improve the collision probability, the collision efficiency for fruitful infection. And many of these challenges can be basically, there we go, thank you. They can be overcome by using polyvalent phages. What I mean by that are phages that would have broad host range, that have the capacity to identify receptors on the surface of many different types of bacteria, from saccharides, and therefore they can recognize and attack multiple hosts. And if we were able to use cocktails, that is, mixtures of these polyvalent phages, you could have a very broad range to attack bacteria. And on top of that, you could grow them on fast-growing benign hosts and not have to worry about the rather expensive purification of phages by separating them from pathogenic bacteria. So we developed a method to isolate. By the way, we know very little bit about polyvalent phages because the methods we use to isolate phages are unintendedly biased to selecting narrow host range phages. But what we did was that we would have hosts of interest and create lawns and grow phages that we get a pool of them from activated sludge or from the ocean or from soil. Maybe you have 100,000 different phages. Maybe only 1,000 grow on that bacteria, on the green one. Those that grow, we pass them to the next host, and the specialist phages stay behind. They will not grow on the second host. Maybe out of that 1,000, only 50 grow in the next. And we do that several times, and by the end, you end up with one or two. They're capable of infecting all of these hosts. That's what we call with the polyvalent phages. And just to compare their performance, we were targeting an E. coli that we isolated from activated sludge that happened to have the NDM1 gene that I was talking to you about. As you know, that gene degrades the last line of defense that we have, the lactamase antibiotics. And as expected, this bottom green line did better. A coliphage, a specialty phage that kills E. coli, did better at suppressing the growth of E. coli than our polyvalent phage, which was the blue line, in pure cultures. That's expected. The other one is a specialist. However, when we have a well-defined mixed culture, in this case we add Pseudomonas putida, the polyvalent phage grows on E. coli, but also uses Pseudomonas putida as a host, and it reaches 10 times higher concentration, 10 times higher titers, which means a higher probability of attacking the host, and therefore we get an order of magnitude improvement in the control of the target E. coli. So we used, therefore, this phage to see how we would perform on activated sludge microcosms. And here, we grew it on Pseudomonas putida, which is benign and fast-growing. We didn't have to separate it. We had the whole bucket. And as expected, the polyvalent phage, which is the blue line on the left, did a lot better, one order of magnitude better than the coliphage. And also, after the polyvalent phage have done their job in killing their host, they basically collapse. They follow a typical predator-prey relationship, and eventually they disappear, leaving behind no residual disinfection byproduct. Now, of course, it's very important to consider potential unintended consequences. You want to make sure that the phages don't go lysogen and deliver pathogenic genes, which is easy to do with omics. You just sequence them and make sure that there are no problematic genes present. But also you want to make sure that they don't impact functions that are important, like the removal of BOD, which is one of the main functions. So we conducted some experiments In this case, we use a multiplicity of infection of 10. That means 10 phages for every NDM1 gene. And we saw no effect on heterotrophic activity in respirometry experiment, meaning that at least the BOD removal function is not being impacted. Now, I did mention earlier that one concern for the application of phages is that bacteria develop resistance to them. They may change their surface receptors so that phages don't recognize them. Or they have enzymes that degrade viral DNA or RNA, and that is a problem. But the good news is that bacteria that become resistant to phages, we call them BIMs, bacteriophage insensitive mutants, BIMs, right? They grow slower, 30% slower. That means that they become easier to outcompete by other non-resistant members and easier to eradicate when you waste biosolids. So that is a good news, but also, interestingly, bacteria dump plasmids that are not needed, and in the absence of selective pressure by antibiotics, the fitness cost that they have by being basically resistant to phages accelerate plasmid loss to alleviate the metabolic burden of carrying those extra genes, that extra luggage that is not needed. So with the phages, not only you get fewer antibiotic-resistant bacteria that are left present, but those that are left tend to have fewer antibiotic resistance genes per cell. Now, looking forward, we all know that there has been a revolution in omics. There is a lot of advances in microbial ecology, modeling, and in synthetic biology that are going to enable the use of phages for precise microbiome editing. And I'm talking about not just suppressing, but also enhancing some microbial activities that you may care about, as I'm going to illustrate in a little manner. But the idea is to manipulate microbiomes in an ecologically informed manner. So, for example, there we go. If you have deeper sequencing, so that you can get species level identification, genus level is not good enough. And you couple that with some microbial network analysis to understand how bacteria are cross-feeding each other and basically their interconnectivity. And maybe with the help of some machine learning, then you might be able to identify who are the species of interest, who are the ones that are carrying the most problematic genes, and who their friends and who their enemies are, and perhaps promote broad functional shifts. In the top, for example, you may want to indirectly foster the growth of beneficial bacteria. And by identifying its enemies, by enemies I mean competitors, predators, could be a Delovibrio that is preying on it, or amensalistic species, and target those so that your bacteria that you want to help flourishes. Alternatively, in the second one, you may want to suppress indirectly your target, I got it, and the growth of problematic species by targeting its friends. And by friends, I'm talking about symbionts or cross-feeding species. Let me illustrate it with an example that we just conducted. If I can advance the slide, that is. I don't know if it's the battery, but can you help me manually advance it with your hands? Thank you. So in this example, we're targeting a Porphyromonas gingivalis. This is a bacterium that is becoming a great concern due to its growing antibiotic resistance, and it causes periodontal disease. It essentially releases gingipains and cytotoxic fatty acids like propionic acid and also butyric acid. And essentially, this is a bacterium for which never has been a phage isolated. It doesn't seem to be susceptible to phages, which is a big problem. However, thank you very much, we're lucky that this bacterium cannot attach to saliva-coated surfaces. In order for this bacterium to form a biofilm, it needs the help from an accessory pioneer colonizer, which is called Streptococcus gordoni. And it happens that recently there was a phage that isolated that can kill gordoni isolated in Japan. So we tested it in well-defined biofilm cultures to see if it could help alleviate this problem. And as you, And as you can see here, as expected, the phage that is targeting the pioneer is very effective in indirectly suppressing the attachment of the target, which is the gingivalis strain. And as expected, with less pathogens present, that significantly decreases the production of the cytotoxic fatty acid, propionic acid, which are the yellow lines, so that it worked really well. Now, of course, we would need much more complex cultures in the more realistic conditions to delineate the merits and limitations of this indirect phage control approach, but this proof of concept is suggested that we might be able to expand the applicability of phages to suppress antibiotic resistance bacteria and enhance public health suppression. So last slide here, right? We have seen that some wastewater treatment plants can definitely serve as breeding ground and point sources for superbugs. And effluent disinfection may not reliably mitigate their discharge, either in biosolids or discharge to rivers. So it's going to be up to us to contribute technological innovation to basically improve disinfection approaches, but also apply ecologically informed editing to the bioreactor microbiome, perhaps using phages to hinder ARG proliferation, but also we need to rethink the way that we build and operate wastewater treatment plant in a manner that exerts selective pressure for bacteria to dump their resistant genes. With that, I just want to thank the NSF through the PIRE grant that Peter Vickesland is the PI for, that funded a lot of this work. And I want to thank here the students that generated the data, and I'll be happy to answer any questions.
Thanks, Pedro. Eddie Citrin from the Volcani Institute in Israel. I was wondering, you sometimes see, and I think in your data as well, that you reach sort of a minimal load of bacteria, which afterwards the phages don't seem to be able to act. So how, and it seems quite high, around 10 to the third, right? So how do you deal with whatever is left over and trying to eradicate the low abundance type of more pathogenic strains?
Yeah, that is true. A lot of times phages are not going to remove 100% of the bacteria. My hope is that they will remove sufficiently so that your immune system can cope with that and not be overloaded. But I see phages as just one item in the menu of solutions that could be used in conjunction, either simultaneously or sequentially with other control approaches. And also rotating phages with other antimicrobial strategies might also be a good idea. Thank you.
Okay, Jianhua Guo from University of Queensland, Australia. Very fascinating research. And I have two questions, and one is a specific and one is a general one. For the first one, have you conduct a sequencing to confirm your polyvalent bacteriophage? So why they can infect multiple host bacteria? Did you identify the receptor for that bacteria host? This is my first question. Second one, for the wastewater effluent, we do have very diverse ARB, right? Very, very diverse. So how do you do that treatment? You want to do the cocktail, or so how do you target specific ARG or ARB in the wastewater effluent?
Okay, regarding the first question, I have not identified the receptors. I tend to be more on the engineering side but however, there are programs out there from the University of Vienna that just using omics data can predict the host range of phages, and they seem to be working really well. Yes, of course. So the first thing is you need to figure out who are the keystone species, among other things, that you may want to disrupt, and who are the major carriers of these ARGs. It has to be omics informed. And you need to select, you need a bank of phages that know this one targets X, Y, and Z, and then combine accordingly to what you have. It's not going to be, again, 100% effective, but I'm hoping it will help, and it deserves further study.
Yeah, thank you.
Our next speaker is Michael Parsi. He'll be talking about phage science, therapeutics, and research, and the Public Health Agency of Canada's new phage therapy program.
Good afternoon, everyone. My name is Dr. Michael Parsi. I'm the head of the new Phage Science, Therapeutics, and Research Unit with the Public Health Agency of Canada. And so this is a new program that's coming into place as a phage therapy program in Canada. Before I begin, I'd like to acknowledge that I live and work in Treaty 1 land, and I actually grew up in Treaty 2 and 3 territory, which is the traditional territories and ancestral lands of the Anishinaabe, Anishinaabe, Cree, Lakota, Dakota, Dene, as well as the Red River Métis peoples. I have the privilege of going after Dr. Alvarez, so I only have to do a quick synopsis of bacteriophages and their specifics. So they are viruses that infect different bacteria. They are incredibly specific in most cases, and the phages that you'd want to use in phage therapy are called lytic. So they do not integrate into the genome of the bacteria that they're infecting. They only go through a replication cycle. And this replication cycle ends in lysis, which is rupture of that cell. And so leveraging that process and using bacterial phages to infect hosts and cause this lysis effect is called phage therapy. Phages have some fantastic attributes to them. They are mostly biocompatible, so they have minimal impact on the immune system, but also they do not impact the microbiome. And so by using phage therapies early in a treatment process, you can actually leverage the microbiome that's present to put pressure on the pathogen and remove it and sequester it within that process. And so phages have really undergone kind of a renaissance as AMR has increased. So over the last couple of years, there's been phage institutes pop up across the country, across US, as well as organizations such as Phage Australia that are starting to use these technologies more often. So the PhageStar program is a 5-year pilot project, which is going to establish a centralized phage collection within the NML in Winnipeg, as well as provide a phage susceptibility testing service for Canadian clinicians to access. So this program and this product is kind of the brainchild of Dr. Michael Mulvey, who some of you may know, and it's something that he was able to put into place before he passed away last year. So what he saw was that we had all of the pieces that we needed to put together a great phage therapy program within the NML. We have great core facilities for genomic sequencing, we have robotics, we have an antimicrobial susceptibility testing service already in place that we are able to model our program after. And with that, we also have an extremely large antimicrobial resistance collection through programs like the Canadian Nosocomial Infection Surveillance Program, which regularly monitors hospitals across Canada for AMR, and so we have those isolates to work with. We also have a number of environmental programs as well, such as CPARs, but also our wastewater surveillance programs that have come in over the COVID period. And so we actually collect wastewater from across Canada on a regular basis to perform surveillance. And so this concept of having a large AMR collection and also essentially weekly deliveries of environmental samples from across Canada really gives us a lot of leverage to do things like phage isolations. So in terms of timeline, this is a project that just came in. It came in at the end of 2023. I started with the program at the end of September. And so right now we're really working to recruit into the program, create our dedicated lab space, and all of our initial contacts for bringing phages in. We've just moved into our second year, which is the establishment of the collection. So we are working with our collaborators for acquisition of the phages. And then this is when we're going to start our characterization process for genomic sequencing, et cetera, that I'll talk about a little bit later. But also validate the protocols that we need to put in this service. And finally, we're working to develop a third-party agreement so that we are able to send out our phages to be purified and quality controlled before they're delivered to the clinician as a therapeutic process. Within the third year, we're going to soft launch the program. This is when we are going to start accepting requisitions through our guides to services that we have set up with the Public Health Agency. And our target goal is to treat 4 patients within that year. And this is a relatively low number, but we're really hoping to have a really comprehensive follow-up process, which is something that's been missing from a lot of phage therapy trials that have been occurred in the past. The last 2 years are really about upscaling. So we're going to be focusing on expanding the capacity of the lab, supporting new pathogens, new patients, seeking funding to do some of the research activities I'll discuss at the end here, and then finally evaluating the program and implementing some form of it in a long-term basis. So to create the Phage Star collection, we are looking at two kind of avenues. We have the isolation process. So we have those large number of environmental samples in AMR, which I've already discussed. But we're also working with a lot of academic collaborators. And the goal is that we are a public lab, and therefore we are not interested in things like monetization. And so we can guarantee through our IP office that we will not be sharing phages. They will be used exclusively for phage therapy. They will not be used for research without consent or redistributed. Within this process, we're also isolating new phages, and we're hoping to be able to distribute these phages through the Felix Durrell Reference Center for Bacterial Viruses through University of Laval. And that way that we are able to promote open science and access to materials that we're isolating. From a characterization standpoint, there's two avenues. So you have your phenotypic, which you're looking at the host range, which phages infect which hosts, potency, so how effectively the phage is able to knock down that host, and then the maximum titer to see if it can actually grow to a certain level that's going to be therapeutically relevant. On the genotypic side, it's mostly just whole genome sequencing and doing an evaluation from there. So we're looking to identify genes that are related to toxins, lysogens, so phages that are able to have, or that have recombinases or integrases that would potentially suggest that they would integrate into the host genome, something you want to avoid for phage therapy. And then lastly, anti-phage resistance mechanisms. So there's a CRISPR system, which is a resistance mechanism for phages, there are anti-CRISPR proteins that exist within phages that actually provide them resistance. And so identifying those phages in advance can really promote the turnaround time for a phage susceptibility testing service. But really the goal is that turnaround time and also to pre-certify the safety of the phages that we're working with. In terms of the testing service itself, we're developing the screening service so that clinicians are able to essentially send us pathogens that we will be testing. We will then be managing that third-party interaction for purification and quality control to bring it into a therapeutic state. And we will, of course, be collaborating with our partners over at Health Canada to ensure that the phages can actually be delivered in a reasonable timeline. And we're really hoping to establish really strong channels of communication with Health Canada so that we're able to push these out very quickly. In terms of phage susceptibility testing, each lab really kind of does it differently, and there's no international standard for what phage susceptibility testing really means. As I was coming into this program, there's a paper that was put out by the Israeli Phage Center in hopes of standardizing this process, which is the figure on the right there. But in all of these processes, they have three main components, which is phage propagation, phage potency, and then phage synergy and antagonism, which is essentially to look at the antibiotics the patient's already getting and the phages you hope to give, and make sure there's no negative interactions there and that the phages aren't compromising the antibiotics, or by adding multiple phages together, you're not compromising the effectivity of one of them. There are three main components to a phage susceptibility testing program at a basis, which is spot tests, which is using unconcentrated or high concentrated or undiluted phages to quickly test on a bacterial lawn if you have an interaction. And so this is just a large-scale screening process that you then break down into efficiency of plating, which is to look for plaques within those plaque formation to determine how many phages are being produced and what the replication rate is relative to a reference strain. And then an optical density read is very simple. It's just looking to see if you have growth suppression of that bacteria over time. Where I'm really hoping to bring the program and kind of accelerate phage susceptibility testing is with in silico phage host range prediction, and as well as molecular quantification, which are my backgrounds. So there's been a recent paper that came out for phage susceptibility testing and host range prediction for E. coli and UTIs. And so looking at that left panel, those are multiple phages along the bottom. Red, it did not infect. Blue, it did. And then so looking at the middle panel, that is the AI model that was used to predict. And so after, if you have less than 60 or so hosts, it's not going to predict accurately. At the very end, there's one called CHAP1, with those blue are boxes at the end being true positive and true negative predictions for host range using AI. And so overall, if you look at those two groups and you don't think about those categories, you can still see there's two camps there, one that infects, one that doesn't, top and bottom. And so even using this as a prediction method very easily, you can determine which phages you actually need to screen and not screen with a very high level of accuracy. So in summary, the Public Health Agency is putting together a centralized phage collection. We are establishing a phage susceptibility testing service. We are going to be managing the preparation regulatory aspects of that and moving into genomic and molecular quantification. And just before I end, I will say the reason I was really happy to present here is that to hopefully stimulate the discussion that there's a lot of phage technologies that are coming into place, and keep that in mind as you're having your discussions, that there's going to be environmental sources, new phage applications in animal feeds, in disinfectants, in the healthcare system, and all of those things are going to have an impact on what we do going forward. And with that, I'd like to thank you and answer any questions.
Hello, Thibaut Stalder in San France. I had a question about, you mentioned the interaction, like you're looking at the interaction between the antibiotic resistance and the phage when you use them. I was wondering if you observe a lot of collateral sensitivity where development of a resistance to a phage would restore the sensitivity to an antibiotics. Is that something that happened?
It really depends on the species you're working with. So something like Pseudomonas, one of the phage targets is a efflux pump called AMR. So in that case, the resistance that, or the development of resistance within that bacteria to the phage actually reduces that efflux pump and then restores sensitivity. But that's not always the case. It's really dependent on what the target of that phage is and if you're deregulating it by adding in that phage.
Okay, thank you.
Our next speaker, Shanshuo Lin, and she will be presenting phagotrophic protists preserve antibiotic-resistant opportunistic human pathogens in vegetable phyllosphere.
Good afternoon, everyone. My name is Shanshuo Lin from Institute of Urban Environment, Chinese Academy of Science. And today I'm very honored to have such an opportunity to share our recent work about the products can preserve antibiotic resistance opportunity pathogens in the vegetable phyllosphere. Throughout our history, humanity has always defended against all kinds of pathogens and infection diseases. And until we find the antibiotic, it really gives us hope to win this war. However, the misuse of antibiotic also provide new global health challenge with resistant bacterial, complicated the treatment and make them ineffective. So as we confront these colleges, it prompts us to think that it's a good, better understanding of the fate and destiny of viral factors or this antibiotic resistance can help us to minimize these challenge issues. So we try to ask questions about when and where do they come from and why they persist and how. So about when and where questions, yesterday Professor Wright has provided us a lot of information about it. So he said that, yes, the antibiotic resistant is an ancient and natural phenomenon. So it exists and has been prevailed during the pre-antibiotic era. So we wondered at that time what drivers or what's the key factors can push them to nowadays. In the natural environment, bacteria face with a lot of stressors like abiotic stressors, the changes of environment conditions, also the biotic stressors like the competitions for resources or the predation pressure. And to fight against these challenges, the bacteria have to develop their own defense mechanisms. But unfortunately, some of these mechanisms may do harm to our health because they can serve as viral factors when they infect human. And some of them may contribute to the spread of ARGs. So all this evolution or this emergence of ARGs or the viral factors, we think they might because the bacteria need to survive in this environment. And as I mentioned, the predation is one of the important factors to the emergence of ARGs or viral factors. And the protozoa is one of the major killer of bacteria in the most of terrestrial or aquatic systems. The protozoa is belong to the protist, which has about 1.5 billion years of evolutionary history, and it contributes to the 5 gigaton of carbon to the global biomass, and it plays a very important role in the nutrient cycling or the turnover of microbial energies. According to their functions, they can mainly divide it into 4 groups, such as the parasite and phototrophy, like some of the algae, they can prefer photosynthesis. And protozoa are one of the big group of them. They can consume bacteria, fungi, and also even other protists or the nematode. So they do exert strong predation pressure to the bacterial communities and can shape their compositions or also their functions. So faced with such a strong enemy, as I mentioned, some bacteria can involve the defense strategies so that they can survive and even replicate within the protist. So some of these pathogens is hide within this protozoa deliberately because they want to avoid the environmental stresses. So at this time, protozoa can act as the Trojan horse to provide a protective niche, and also they can carry this endosymbiont to travel around, and if the endosymbiont can find the suitable place, they can return back to the environment again. And also, some articles says they can function as the multi-parts or chaining grounds. Because protists can be a place where the intracellular pathogens or bacteria can exchange their genomic information to help them each other to survive within this environment. Some of the pathogens can even be more hypervirulent after passing through the protozoa, like amoeba. So we do believe that a good understanding of protozoa and bacterial interactions, their associations, will help us to understanding the ARG or the viral infectious issues. But the thing is, nowadays we do pay little attention to this kind of fact. We mostly focus on the human activity, the contaminant impact to the ARGs, and the previous study do a lot of great job with these the module strains of bacterial and the module strains of protozoa, but they mainly do experiments in virtual conditions. So we think we need robust evidence to know how the risks of the pathogenic endosymbionts within pathogens, and what about the pathogens and bacteria and protozoa interactions. And considering that consuming raw vegetables is a healthy lifestyle for the youngers and consumers, the vegetable philosopher can also harbor various kinds of also AMRs and including pathogens, and it is a bridge which can link the environmental microbe to our human microbes just through the food chain. So we collect vegetable samples from 4 major supermarkets in Xiamen, China, and these include 4 different vegetable species that can be eaten raw. For most Chinese, I don't know if you eaten them raw. And some of them are conventional product, and some of them are organic product. And first we look at the proteins in these leafy greens, and we find that the copoda, the yellow bars, is the dominant protein almost across all the samples at the genus level. And as for the function group, phototrophy or the phage-trophy, which is also called a protozoa, share the similar relative abundance. And the alpha diversity or beta diversities of protozoa communities differed across the vegetable host. And then we quantified the human pathogen marker genes through high throughput qPCR method. This method including 68 primers detecting 33 human pathogens. And we find that a total of 26 potential pathogens was detected in 90% of the samples, with a horrible number. And among them, the Klebsiella pneumonias was the dominant pathogens. And then we try to find the associations between the microbial communities and these kind of pathogens, marker genes, use the statistic methods. And we find that the total abundance of these marker genes are positively and significantly linked to the protozoa. And the protozoa is also the best predictor rather than the bacterial or the fungi. They could best predict the relative, the absolutely abundance of some of the human pathogen marker genes. And then we find one of the key protists, which is belong to the copoda. It is very closely related to the human pathogen marker genes. And we wonder that whether this kind of close relationship what's behind these statistics relationships. So we isolated some of the protists in the vegetable philospheres, and luckily the copoda was included, and we isolate a total of 11 protozoa. And then we just lysis these protozoa and obtain the endosymbionts using the whole genome sequencing. And we got 36 the high quality genome information, and 34 of them is reported to the human pathogens. And most of them both carry ARGs and viral factor genes and metal factor genes. And we find that all these endosymbionts carries ARGs, especially the most dominant is the multidrug class. And we find positive links between the number of ARGs or VFGs, also the MRGs. And this positive link we think that this positive link may explain why they can survive in the practice, and also it will pose a threat to our human health, because it may cause the core transfer of these ARGs, VFGs, or MRGs within this endosymbiont. So to conclude, we find evidence that the philosopher protozoa can preserve these antibiotic-resistant opportunity pathogens, and the proteus is key indicators. And we find the key one is Capoda, and we find the core occurrence and the potential core transfer of ARG, VFG, and MRGs within the endosymbionts. And finally, but we also have a lot of things to do in the future. We need to explore more, the more endosymbionts. And that's, I'd like to say thank you to my supervisor, Professor Yongguan Zhu, and our team. Thank you.
So I have a very simple question. Does the washing with only water will eradicate or remove this protozoa from the fresh produce, or does it require any sort of specific treatment?
And you mean we need another method to remove this protozoa?
So if this protozoa in the fresh produce, and you said in your last slide, wash thoroughly or carefully. So washing with only water will remove the protozoa from the fresh produce?
Yes, yes.
Only with water, right?
No matter what it is, but wash them can remove the most of the protozoa as we do some experiments. Yes.
Thank you.
Hi, Nicholas Ashbold, University of South Australia, Australia. My question is in relation to, you talked about these symbiotic bacteria inside the protozoa,
Yes.
but there are also amoeba-resisting bacteria that end up killing the host and spreading themselves further and being taken up by more hosts. Have you looked at some of those amoeba-resisting bacteria?
We do not use the in vitro experiment to verify their endosymbiont relationships. We just look the genotypes informations.
Because it's been shown with other protozoa bacterial relationships that some of the genes from the symbionts are shared with those that end up killing the host. And so there's another potential avenue for transmission of these genes.
Yes, there may be some core evolution between the protozoa and the endosymbionts, but in this work we do not care about these scientific questions. So maybe in the future we will more focus on the core evolution.
Thank you.
Thank you.
Our next speaker is Heike Schmidt. She will be talking about protozoa predation, a major pathway of E. coli removal in aerobic granular sludge.
Thank you. And as you can see, I'm not Zhaolu. I'm Heike Schmidt, supervising Zhaolu together with Nora and Mark. She's doing her Ph.D. in the Wageningen University, and she's a very, very pleasant personality and also gifted Ph.D. student. So the subject of her PhD thesis is to look at the removal of both pathogenic bacteria and micropollutants in wastewater treatment of municipal wastewater, and look at activated sludge processes, but also another process I'm going to mention in a minute. And as we heard, wastewater treatment plants can be breeding grounds for making new combinations of mobile genetic genes and hosts, but also actually wastewater treatment plants do a very great job in removing basic human-associated bacteria, often by 2 or 3 log across the world in well-functioning wastewater treatment plants. And surprisingly, actually the mechanism of the removal is not perfectly known. We think that bacteria attach to activated sludge flocks, and also we know that predation happens, but the actual contribution of these different processes, their role in different treatment options is not fully known. And that is where Zhaolu started. She also starts on a specific technology, on aerobic granular sludge, AGS. That is a technology that has fewer energy needs, a smaller footprint, and is marketed under trade name of Nereda. In my hometown in Utrecht, we actually just have a very new Nereda plant of about these dimensions. That's also where her sludge was taken. And the interesting thing about the AGS process is that it consists of granules. They grow throughout the process, from small granules to larger granules. And in these granules, you have a highly structured microbial community that serves to run the main functions that are needed for wastewater treatment. So we have a coexistence of small granules and also flocculent sludge and larger granules. And all these granules slightly differ in their physicochemical properties, in their charge and their hydrophobicity, and they also differ in their protozoan community. So the question is here how far these differences also affect removal of human-associated bacteria. So her research questions are actually, what is the main pathway of removal of human-associated bacteria? Is it predation or attachment? Is there any variation between the different sludge sizes that she has in her treatment plant with respect to removal performance and the protozoan communities? And how do these two influence each other? And also, what is the difference between this process, AGS, and classic activated sludge? What she uses is microcosm experiments in which she takes freshly harvested sludge from Utrecht and she separates this in different sludge sizes. Then she adds E. coli, viable E. coli, at a concentration that is roughly equivalent to what we would find in influent. And then she studies the whole process, so predation and attachment together under oxic circumstances, because that leaves the protozoa in the conditions to prey. When she changes the conditions to anoxic conditions, that makes the protozoa inactive, and thereby one can study only attachment. This is also combined with a mini grazing experiment because mere disappearance of the E. coli doesn't prove the process. So what she did there is she took the same sludge fractions and she spiked them this time with labeled E. coli, with fluorescently labeled E. coli in a small tube, and then she studied what she saw by classic phase contrast and fluorescence microscopy. So, to the results. What we see here is the disappearance of E. coli from the supernatant of these microcosms over time. So we see that as a really quite quick process. Equilibrium is reached in some 18 hours. And that is also important because the AGS reactors are run as sequencing batch reactors, so they have a hydraulic retention time of anything between 6 hours and 30 hours. So they are run in batches of a day, roughly. And we see that there's quite a strong difference between the different sludge fractions with respect to their removal potential. We see that the yellow and the dark blue ones, these are larger granule fractions, are actually able to remove E. coli by more than 3 log units. So that is a significant removal. This is what's shown here. So it's the middle-sized and the larger granules that actually do the best job, have the greatest removal capacity. If we look at attachment only, this is pretty moderate in these sludge fractions. So under anoxic conditions, we see very little removal, and we also see less of a difference between the different sludge fractions. And I think you have to believe me, it's not really visible that if you go to lower sludge sizes, attachment removal increases just a tiny little bit. So, can we prove that this is actually removal by predation, or is that some artifact we have included in our arguably in vitro systems? First of all, something else. By running these experiments for attachment and predation, and for attachment only, we are able to derive the fraction that is removed by predation by simple subtracting the two. So, subtracting the conditions anoxic and oxic. And this is what you find. So you find that indeed in the middle-sized sludge fractions around 1 to 4 millimeter, you see 2.5 log removal. So that's actually pretty significant. So predation indeed can enhance E. coli removal. And I forgot to mention E. coli here serves as a simple model organism for human-associated microorganisms. So predation indeed could be a crucial pathway in at least AGS systems. And it's the middle-sized granules that act more aggressively prey, although it's not, of course, the granules that prey, but the protozoa that we'll see in a minute. And actually, the granules overall, they do better than activated sludge, but the smaller granule fractions have about the same efficiency as activated sludge has. So, now, at least for me, to a new branch of the taxonomic tree, what we see here is the range of ciliates and protozoans we observed in the microscope. So what we see on the left-hand graph is the phase contrast microscopy view, and on the right-hand side with the fluorescence coupled in there. And I think it's convincing that you see that indeed the labeled E. coli, they migrate into the protozoans. They are located within the vacuoles of the protozoans, and that actually is active predation. And that is in contrast to anoxic conditions when we don't see that movement at all. And what we see here is some examples of so-called sessile ciliates. They have stalks with which they are connected to their granules, different species, Epistelis and Vorticella here. But there's many more. There's crawling ciliates, there's free-swimming ciliates, and there's also testate amoebae, which we just heard about with respect to Legionella. And we saw that many of those are able to accumulate E. coli, but not all of them. So in this case, this specific amoeba was not really convincingly accumulating and preying on E. coli. So, do the protozoa differ in the different sludge sizes? Yes, they do quite clearly so. So we see that the larger sized granules are dominated by the dark blue species that are sessile ciliates, and we see the amoebae and the crawling ciliates only appear in the smaller sizes. And also some free-swimming ones. And if we move into the sessile protozoa, we also see a shift in their species composition from Epistelis in the large ones to more Vorticella-dominated sessile protozoa in the smaller fractions. So more Epistelis in the larger granules, more Vorticella in the small-sized sludge. So, learning from this, we thought that maybe Epistelis indeed may have a key role in preying on E. coli, whereas Vorticella is maybe more active in activated sludge, but not so efficient on preying. And that's what we try to confirm through simple linear regression. So if you regress the predation rate that you observe in the different sludge fractions on the absolute abundance of these species in this sludge fraction, you see indeed with higher Epistelis percentages and numbers, you see an increase in the predation rate, whereas the opposite is the case for Vorticella. And you can also do that in a multiple linear regression that still points to Epistelis as maybe a positive acting species, but this is a bit limited by that co-correlation. So actually I think it's a guess that Epistelis is it, but we can't prove this through multiple linear regression. So from here, we dare to conclude that Epistelis possibly contributes most to E. coli removal, and that it might be interesting to further investigate which actual species are responsible for the greatest part of the removal we see in this interesting system. So, to take home, so we think that predation can indeed be a main removal pathway, at least in active aerobic granular sludge. For activated sludge, it's maybe about equal between protozoa and attachment. The middle-sized, larger granules have the largest capacity, at least, for E. coli removal, and there's very clearly differing protozoan communities, of which the sessile ciliates are most probably the dominant protozoa and also the ones with the highest predation capacity. Vorticella is the one that is more present in activated sludge, so important to study for functioning of a normal treatment plant. And so maybe that leads the way to inquiring whether protozoa could be used, or optimization in the protozoan community, or optimizing their attachment behaviors could be used as an alternative method to improve wastewater treatment. But of course, as we just heard in the talk before, and as we discussed before, protozoans don't only remove E. coli. We don't know yet what happens once they ingest the E. coli, because for sure there's the potential role of endosymbionts, of survival of bacteria within the protozoa. And also, as the bacteria are very close to each other, it might also be possible that there is a gene exchange within the protozoan vacuoles. All processes that need to be studied and are also very interesting and very nice to be studied, as I think. So, thanks to Zhao Lu for working on that, for having her data ready here, and thanks to the team, and thanks for your attention.
Thank you, Heike. Didier Oquet from France, Besançon. Great talk. Perhaps it's a commercial secret, but do you know what the granules are made of?
I must admit I don't. No, but I think it's not so much the composition of the material, it's more that very, very quickly they are colonized by bacteria and by EPS layer. So very quickly, so it's not the material itself but counts, but it's the cover of the biofilm on them.
Hi, Professor Schmidt. Thanks very much for your wonderful talk. Before my question, I should say the word is very small because Zhao Lu actually is our graduate from Tsinghua University in Shenzhen campus. So just now I sent a WeChat message to Zhao Lu. So, okay, my question is that the protozoa in the granule sludge, so what's the function for protozoa in the granule sludge for the granule sludge structure or community structure? So what's the function of the protozoa?
We actually haven't studied that yet, but it's known that protozoa actually can shape bacterial communities. So that has been shown in research and on genetic basis across different treatment plants. So differing protozoan communities would also prey differently on different consortia and thereby shape the bacterial communities. And that is shown, interestingly, through a temperature effect, because protozoa more thrive in warmer conditions, and that's when they would change composition of bacteria as well. But we don't really know how that actually plays out, so the selection of certain bacteria through the protozoans. I think we're very much at the start of discovering what kind of processes happen when the communities change.
Okay, maybe a following question is that maybe it's very difficult to compare the abundance of the protozoa and the bacteria, but if we compare the biomass in one granule sludge, so is there any data for their biomass ratio of the protozoa and the bacteria?
Oh, again, I must disappoint you because we haven't really calculated the biomass of the protozoa because that's actually pretty tricky. You would have to do through microscopy, which is pretty hard on the granules because they're not flat, they're round. So I think we would have to think about, very careful methods to come up with biomass estimates indeed. But good question, yes. Thanks.
Okay, thanks very much.
Yeah, hello, Heike. Thank you for this interesting talk. Alexander Kirschner from Medical University of Vienna. I have one question. You did not consider phage predation, phages in these models. Is this not necessary to do so, or what was the reason for that?
You cannot do everything at once, right? That's the simple reason. Of course, it would be interesting as well, yes. And but I think for phage, that would be really artificial because we are only spiking with E. coli, right? And for phage, you would be interested in their effect on the whole bacterial community. But I think for the protozoan, it's also interesting to look at whether they actually feed selectively, but we haven't come to that yet. But I think that might be the great difference between phage and protozoa, because protozoa, at least potentially, are less picky on what they prey on than phages are. Yeah.
Okay, thank you.
I will invite Shugan Yang to present paper-based microfluidic device for the detection of antimicrobial resistance genes in drinking water.
Thank you for the introduction, and I firstly apologize. I just speak on behalf of my students. So as you know, we talk a lot about antimicrobial resistance, and especially it's kind of the contaminants for the drinking water. And also the WHO also say that the access of the clean and safe drinking water is kind of the sustainable development goals. And also there is a lot of policy has already put out as a government action. For example, in the UK, they put the 5-year program on the AMR action, and also coming up after 2024, the new 5-year actions is really important from tackling these challenges in terms of from the source, pathway, and the receptors. As you you may already know this theory in order to control these contaminants as a global public health. However, I think today I'm going to talk about this new monitoring method, especially rapid and low-cost monitoring method, which can enable this detection in most of the global source countries, especially in the resource-limiting areas, as this kind of the bigger source of this transmission of this antimicrobial resistance. So that can all give you another answer in terms of to get a dynamic monitoring or real-time monitoring or a continuous monitoring of this transmission from the environment and better understand how it is evolution as this antimicrobial resistance. So I'm going to talk about some new technology which is using microfluidic paper-based analytical device. So this kind of the device is from traditional paper microfluidic technology, which is using the pump to drive the liquids go through the sampling and the tubuling, then do the proper sample preparation. We also knowing as it's a lab on a chip. Here we moved this concept from a lab on a paper, so which is using easy, low-cost paper to manipulate these liquids and the samples in order to do the proper sample preparation. Then you can do the sample preparation and the purification even in resource-limiting areas because it doesn't need any centralized machine or equipment or any power. So there is a couple of technology to fabricate this device. So mostly the concept is they're using the wax printing to generate this wax printer in order to get the wax hydrophobic layers to confine these liquids from one panel to another panel. Then it actually can go to do the proper sample preparation. So everything we can run on this lab on a paper to replace which you can be doing on the centrifuge or some power-empowered facilities. So I'm going to give you some more details about that. When talking about this paper microfluidic technology, we want to really mention Professor J.J. Whiteside from Harvard University. They published a very impactful paper in 2007 using the screen printing technology to fabricate electrodes, which can replace the traditional gold electrodes to monitor the database for monitoring the glucose concentration. So that's really make a lot of application in these areas in order to create some low-cost sensors or even but can test these contaminants, including AMR, in the field. So, however, what our doing is slightly different. So we fabricate this paper microfluidic device using wax printing technology. As you can see that there is, actually there is three components of this device. First is wax printed chromatography paper. That's enable us to do this hydrophilic and hydrophilic layers. As you can see, that you can easily design and fabricate these patterns. And then we can, secondly, we have the glass fiber. So glass fiber is a material which can absorb the negative charged nucleic acid. So by this adsorption or absorption, we can actually purify this nucleic acid from the either water samples or clinical samples. So in this case, we can purify this nucleic acid from these complex environmental samples without any power, without any centralized facilities. So the third part is a plastic plate. We can accommodate this amplification to isothermal amplification. For example, we can use loop-mediated isothermal amplification, which does not need thermocycling, but we can accommodate it within the constant temperature to amplify this nucleic acid in order to identify this presence or absence of these pathogens, or even we can do the calorimetric detection for this quantitative detection. So eventually, we can using the other calorimetric change or using lateral flow device to quickly give you the detection results. So that's kind of the first concept of this device. We initially developed this for the veterinary diagnosis and tested it in a local Indian farm for demonstration of the testing semen sample for field test for the capability of field testing. And later on, we further changed the design of this device by introducing enrichment samples. Unlike clinical samples, we only need a drop of the liquids. For example, it's only microliter scales. However, when you analyze these environmental samples, such as drinking water samples or even wastewater samples, normally we needed 50 milliliter or 200 milliliter. That sample size is different. And another challenge is those of virus, bacteria always present at an extremely low concentration. As you can know, for some of the detection for flow cytometry, they may need 50 liter or several hundred liter to concentrate in order to run through the flow cytometry detection, which has already been widely used in the water utilities. So that's caused the extremely challenging in terms of the economic and labor. Here we're using a easy syringe-enabled filtration system to enrich this nucleic acid within probably 50 milliliter to 200 milliliter of these pathogens. And then we imagine we're using this paper-based device to do the proper sample preparation. and then to purify this nucleic acid from those drinking water, and then we can manage to test them using isotope amplification or isotope amplification coupled with CRISPR-Cas or some of the biotechnology to amplify this nucleic acid. So here we also introduce a new design by introduce high throughput panels by folding and unfolding. So that's enable us to detect multiple pathogens in single device. So we also, by using these two technology, one is a sample enrichment technology, another is a pepper-based detection. We actually can detect those pathogens in drinking water on the site, or even in the highlands, for example, in Scotland, or even in resource-limiting areas in Africa. So this potentially contains a lot of antimicrobial-resistant genes, which cause potential risks. So here is some of the optimization of this device. As you know, this is really using some fluidic technology. The length, the width of this hydrophobic layer can really have effects of the DNA yields. So we want to maximize or recover all the nucleic acid from those environmental water samples. So then we can do the proper amplification that enable us to do the highly sensitive detection. So the sensitivity and the specificity are always challenging for this detection. And also, we, in order to do the colorimetric detection, we replace the fluorescent dyes using a colorimetric dye to enable the naked eyes enabled detection. So that's kind of as a lot of optimization in order to enable this detection with a required sensitivity and a specificity. So for this example, after optimization, we use this detection to test the 3 gene ARGs. And as you can see that we, based on these optimization results, we can achieve the promising sensitivity and specificity. So this is kind of the results. We test them in the real samples. And the limit of detection is kind of to reach around 10 copies per microliter, which is really comparative within the gold standard PCR-based method. However, this detection technology doesn't require any facilities or even centralized equipment, and even without any well-trained technician or specialized. So that's enable us to detect them whenever you want, wherever you can. So this kind of the part of the work for the drinking water detection, but also we also do some extra work in order to really answer the transmission of this ARG, because as you know, most of molecular methods, including PCR or sequencing, detection target is actually the nucleic acid that cannot answer your live or dead. So if it's just detecting the gene sequence or fragment, we develop a new method to identify this live or dead virus. So that's before you try to work around if there is any antimicrobial resistance or not antimicrobial resistance. Especially in drinking water, if you detect a positive, even within PCR-based method, but it's probably just dead virus of this gene sequence that cannot indicate any risk. So that's kind of where we want engineering this detection method onto the paper microfluidic platform. So that's enable us to do more detection. So we also do some of the work for ARG in the drinking water. So I think I just being requested. So at the same time, we are lucky to get funding. Just to briefly introduce this project, we are have UK BBSRC funded a UK Canada project to tackling the challenges of AMR in the management agriculture. Finally, it's kind of open discussion. We try to establish or develop some new detection method, including low-cost sensing, to monitoring these contaminants and new emerging contaminants. But I really want to using multidisciplinary technology to address these challenges and using like new models, including machine learning or new complex models, in order to build up these early warning systems to tackling these global health challenges. Thanks. Finally, thanks for your attention. Thank you for your time, and especially for the fundings. Thank you.
So, Jugen, just a quick question. How do you address non-specific amplification with your LAMP assay?
Yeah, that's a very good question. So actually, it's mostly depending on your, firstly, we need to find the proper gene targets. So we need to get a lot of informatic searching and survey, find the conservative regions, which is very specific. Probably find the species-specific sequence. And secondly, we need to really design the proper primary sets in order to do a lot of, and then following up with a lot of optimization. We can actually compare several species in order to check if there is some overlapping and also specificity of this. But it involves definitely a lot of optimization.
Thank you very much for that presentation. I just want some clarity because you spoke about you having to, differentiate between life and death. How's that done?
So the principle is we call it a molecular viability test. We test the microRNA, which is from the live bacteria, and get a ratio between this microRNA and the genome sequence, so to work out if they have been this alive or dead. So if it is a live bacteria, they will produce this microRNA. If not, they won't. So that's the concept. But we actually make difference to engineering this detection on a paper microfluidic device.